and I work at Lund University Clinical Research Center in Malmö, Sweden, and the presentation will deal with type 1 diabetes, viral etiology, and autoimmune pathogenesis. My disclosures are shown here, and I want to start off by indicating the frequency distribution of newly diagnosed type 1 diabetes children in Sweden. It's 11,000 Swedish children diagnosed between the years 2002 and 2017. And as you can see, most of the children are diagnosed with the disease when they are between 10 and 13 years of age. But there are already children who are diagnosed with type 1 diabetes at 1 year of age. Rarely, though, are children diagnosed before 9 to 12 months of age. The prerequisite to understand this disease development is the genetic etiology of the disease. Since the 1990s, HLA-DR3, DQ2, or DR4, DQ8 haplotypes are known to be necessary, but not sufficient. So, for example, as shown by Sanjeevi and co-workers already in 1995, 90% of newly diagnosed type 1 diabetes children, representing about 26% of the Scandinavian population, had either DR3, DQ2, or DR4, DQ8. The primary function of HLA molecules is to present foreign antigens to elicit T-cell responses. The number of HLA heterodimers expressed on the cell surface of antigen-presenting cells is directly related to the range of foreign antigens the host can present to T-cells. HLA presentation of autoantigens may cause cell-specific autoimmune disease. And this is important for this presentation, that self-reactive CD4, CD8 positive T cells, or primarily B cells producing autoantibodies, are markers for a self-reactive response as opposed to a reaction protecting us from an infectious agent. So the second prerequisite to study the etiology of type 1 diabetes is autoantibodies as biomarkers. We have 4 autoantibody biomarkers in type 1 diabetes: GAD, IA2, zinc T8, and insulin autoantibodies. And recently there is a development to approve these autoantibodies as enrichment biomarkers in type 1 diabetes prevention clinical trials. And the European Medicines Agency have already completed a public consultation, and an application is being prepared for the FDA. The etiology question is, what is the trigger of a first appearing autoantibody? And the first appearing autoantibody is the endpoint in investigating the etiology of type 1 diabetes. Smaller studies listed here have tried, but none of these smaller studies had enough statistical power to answer the question. The National Institutes of Health, NIH, in the US provided funding for the TEDdy study, and that was designed to provide sufficient power. The development of autoantibodies was the first primary endpoint in the TEDdy study. In 2004 to 2010, 440 newborns were screened for HLA-DQ. The genetic susceptibility defined the inclusion criteria, and the primary endpoint was confirmed persistent islet autoimmunity for either one of the 4 autoantibodies. Which one of the 4 autoantibody appeared first? We did not think, although we didn't know, that all of the antibodies were appearing at the same time. By January 22, 878 children have now developed a first islet autoantibody, and 419 have gone on to develop diabetes. The TEDdy study methods are illustrated in this slide. It shows that stool samples were collected monthly to quarterly, monthly for the first 4 years. And blood samples, the red arrows, were collected every 3 months to measure the autoantibodies and a host of different analytes to reflect the possibility to explain why a first autoantibody develops. We collected also other samples, and the parents were interviewed extensively at every visit. The incidence of first autoantibody type was a surprise to us because it was clearly demonstrated in this incidence rate analysis that IAA first, the red curve, appeared very early. The age of seroconversion is shown on the x-axis, and as you can see, the peak of IAA first occurred already at the first year of life. And that was markedly driven by the Finnish children. In contrast, the blue curve shows GADA first, and GADA first started to appear by 12 months of age and then by 2 to 3 years of age, and did not decrease the way the IAA first incidence rate decreased over time. And indeed, the GADA first curve has stayed high, while the IAA first have decreased to very few children per year. The green curve shows the children who showed both antibodies at the same time. And remember, it's 3 months between the blood samples, so we couldn't tell in the green curve whether it was GADA first or IAA first. And as you can see in the table, GADA first or GADA only was 41%, and IAA only was about 39%. And there were occasional children who showed IA2A as the first autoantibody. So when we asked the parents what happened prior to the first appearing autoantibody, the parents gave us the following report, namely that upper respiratory tract infection was reported by the parents 3 to 6 months before IAA first was detected in the blood sample. In contrast, parents reported gastroenteritis 3 to 9 months before GADA first was detected in the blood sample. And this indicates that when virus finally was assayed in this study in Nature Medicine by the TEDdy group, we found that enterovirus B increased the risk for IAA first, and adenovirus F increased the risk for GADA first. And the major finding was shown in this slide, that the risk of IAA as the first appearing autoantibody was if the child had a prolonged shedding of enterovirus B in the stool samples collected monthly. GADA as the first autoantibody were not related to enterovirus B. So the major findings in the Nature Medicine paper was the following, It was the largest known study of eukaryotic DNA and RNA viruses in stools from children ever. Fecally shed viruses were investigated in relation to IAA first and GATA first. Prolonged enterovirus B rather than independent short duration enterovirus B infections may therefore be involved in the development of islet autoimmunity, but not in the development of diabetes in some young children. Early life human must adenovirus C infections were protective, and CXADR, the enterovirus receptor, was found to be independently correlated with IAA first. This study was independently validated by the Finnish investigators, showing similar observation on insulin autoantibody first being associated with the Coxsackie B1 infection. So therefore we conclude that type 1 diabetes is not one disease, it's two diseases reflected by two endotypes. IAA first disease, and the etiology is enterovirus B in DR4 DQ8 children. Insulin, PTPN22 and UBAS3A are independent associated non-HLA risk factors. We speculate that antigen-presenting cells fail to present the virus, but instead present pro-insulin peptides leading to autoimmunity. GATA first disease, on the other hand, may be adenovirus F, DR3, DQ2 children, and BAC2 is an independent non-HLA genetic factors. We speculate that antigen-presenting cells fail to present the virus, but present in GAD65 peptides. And below is shown the incidence curves now up to 12 and 13 years of age. And you can see IAA is very rare as the first antibody in older children, but it's common in the older children for GATA first. Other observations include the metabolomics analysis showing that dehydroascorbic acid at 3 months of age was associated with both GATA first and IAA first, but GABA was only associated with IAA first. And these methionine, alanine compounds produced in the BCA metabolism, as well as fatty acid, also preceded the first appearing autoantibodies at different time points. So other mechanisms may be detected to explain the way by which a virus infection is leading to a first autoantibody. The TEDdy study has been validated by analysis in the DIP study in Finland, where autoantibodies were regularly analyzed from birth up to 15 years of age. And these are the 4 autoantibodies, and they were genotyped for HLA-D or DQ haplotypes. Homozygotes for DR3 DQ2 haplotype had almost exclusively GATA as the first autoantibody. DR4 DQ8 genotypes, on the other hand, had IAA more often with the first autoantibody. And there was a split in the DR3, DR4 heterozygotes between IAA first and GATA first. The two endotypes have different progression in the TEDdy study to onset of diabetes, and you can see that the IAA first seems to represent the more aggressive disease, while GATA first is the milder progressing disease. Also reflected in this illustration there, the second appearing autoantibodies. So if IAA first or GATA first, the child experience IA28 is associated with a more aggressive progression to type 1 diabetes. than if zinc T8, the green line, is the second appearing autoantibody. So the future would include the idea of vaccinate against autoimmune diabetes because it's triggered by a virus infection. And an enterovirus B, Coxsackie, is already under phase 1 and phase 2 clinical trial by Prevention Bio in the United States. The PRV-101 is a polyvalent Coxsackie B virus vaccine, which was licensed from Vactech, a Finnish company, and Prevention Bio is developing as a potential vaccine for acute Coxsackie infection and for the potential of preventing up to 50% of worldwide type 1 diabetes cases. And a similar vaccine will be needed for those enterovirus triggering GADA first. So thank you very much. I'd like to acknowledge my collaborators at the CRC in Malmö. I'd like to acknowledge the Teddy children and their families, our funding from NIH and other funding agencies, and the investigators at the CRC in Malmö. And I also like to thank the TEDdy study research team in the US, Sweden, Finland, and Germany, and the expert laboratories that TEDdy is utilizing for analyzing many samples in the US, in the UK, and in Finland. Thank you very much for your attention.